Boa tarde a todos, eu ia dizer boa noite já. Estou pensando já no happy hour, né? Bem, gente, eu gostaria de começar este seminário, que vocês já viram que é uma organização do Laboratório de Pesquisa em Fármacos, junto com o Centro de Pesquisa Clínica, né, do Hospital Universitário. Eu gostaria de agradecer a presença de todos vocês. O Dr. Gilles já falou que ele está impressionado com tantos jovens, né? E o interesse também. Então, depois o professor Tiago vai fazer uma breve introdução, mas eu gostaria de apresentar, primeiro agradecer também o Dr. Gilles e a Bettina, porque eles estão aqui por conta própria, sem nenhum custo para a gente. Então, o Dr. Gilles, ele é neurocientista, né, formado a graduação na França, em Estrasburgo, e o mestrado dele foi na Universidade de Basel, e o doutorado esqueci agora a universidade. O doutorado que foi em Estrasburgo, né? Então, ele sempre esteve na área de neurociências e ele vai apresentar um tema de grande importância, inclusive participou também de um grupo de estudo do Prêmio Nobel, né, nesta área que ele estuda, né, que é de grande interesse para a modulação celular em termos de câncer e doenças neurodegenerativas. Então, para nós, eu acho que é um grande feito, né, um grande prazer nós termos uma pessoa de grande importância e humilde também, cheia de humildade, para apresentar esse tema para nós. E é esta a nossa função aqui dentro do Hospital Universitário, fazer valer, fazer essas conexões e trazer pessoas que possam contribuir e incentivar também a pesquisa. E o Dr. Gilles, ele é pesquisador da Novartis. Então, ele começou na Novartis enquanto era chamada ainda CIBA, né? Então, ele está há 37 anos, depois CIBA se tornou Novartis, juntamente com a fusão mar de uma indústria. E então, ele é chefe desse setor de pesquisa na área de biologia molecular, né? E também nós já conversamos, há grandes oportunidades de intercâmbio para estudantes, para professores, né, pesquisadores dentro dessa área. Tá? Eu passo a palavra para o professor Tiago, por favor, que ele é chefe do setor de ensino e do HU.
Boa tarde a todos e todas. Então, hoje eu quero agradecer primeiro a presença de todos vocês para poder iniciar esse processo da nossa visita, né, e da nova organização que a gente vai ter em relação às professores e às apresentações. Hoje, o Dr. Gilles, ele vai discutir sobre a parte das doenças lisossomais, disfunções lisossomais, essas relações, e também alguns aspectos em relação à utilização da ração em si. Bienvenue et encore merci beaucoup pour votre disponibilité.
Merci, merci, merci. Many thanks for this really kind introduction. It's a real pleasure. It's more as a pleasure, I must say. It's an honor to be here, because Macapa, as I see, is a beautiful city with a fantastic university. You have all what you need here to do the best science you can find all over the world. And just surrounding the city, there is a forest. And the forest is a miracle. I enjoy Professor Tavares take us 2 days to the forest. And just by staying there 2 days, sleeping one night on the balcony of his houses in a hammock, like a Native American, it was a huge experience. I think this was already starting to transform me, really. So many thanks for coming. And many thanks, Professor Tavares, to give me this opportunity to try to present you what I'm doing now since 5 years actually. No, maybe I have to come back. I have to be used to the system because as simply as it looks here, you see it highlighted by an immunofluorescence. These are lysosomes. They look very simple. They are basically just sphere, just pocket. But when you think about, when you start to study them, you see that this simple bag gets suddenly a planet. A planet because of its complexity, because of its way to control and regulate the size, the growth of each cells in your body. And at least cells constitute your body, your tissue and your body, meaning that this small bag is in the center of your health and your disease. Yeah, I was thinking that before we start to deep dive into the cell biology, I wanted, because it's also the first time that I'm in Brazil, to introduce myself. Professor Tavares and Professor Tiago, they already tell you my way through science. And I just want to add that as the new generation of researchers, of biologists call me, I'm a wet lab scientist. A wet lab scientist means nowadays actually that I make experiments, that I produce data. In contrast to the data scientists that use data that were already produced to make the analysis. So every day I'm in the lab and I'm making experiments. And my background since the beginning is a genome engineer. In the sense that since 37, near 40 years, I touched every techniques that able you to modify the genome. Starting by the homologous recombination in stem cells, in mouse embryonic stem cell, to the nowadays CRISPR, I get through all this kind of technology, and through that, I generated transgenic mouse and knockout mouse that able me to, as you see, touch quite different field of science. But mostly, I was surfing through immunology and neuroscience most of the time, but I touch some other disease areas. You see, my life, my daily life is very rational, very practical, because I have to produce data. I'm paid to produce data. To produce data, you have to make experiment, and in your experiment you dissect, you separate, you differentiate molecules and process. Where my spirit, my soul, my mind is the one of the Native American, in the sense that I see that from the mountains to the river to all form of life, we are interconnected and interdependent in a sacred circle. And I will try, through my presentation, to use this principle, to show you each step integrated in a circle. And we will start with the first circle, the lysosome. Lysosomes are kind of, you see it, a hand row of all the different organelles you have into a cells. This should be a mitochondria, but you have here the lysosome in a cut. And this will be the first circle. We will go through the lysosome, how it's work, and especially what's happened when it's not working. Then the circle will enlarge to one of my favorite project in science, the molecules that are expressed on the double lipid membrane of the lysosome and then transport molecule into the lysosome or export molecule from the lysosome. And then the circle will enlarge to the major physiology in which the lysosome is involved, meaning the autophagy. Autophagy means eating yourself. It's the security when you have nothing to eat. Cells find a possibility when you starve them, when you don't nourish it, to survive. And this is autophagy. And I will try to give you a flavor how important this process is in the normal health condition, but also when you have a problem, when you get a disease. And at the end, a dream, we will go quickly. It's just an introduction, but if you are more interested, I will let to Professor Tavares the more specific review about that, because one of my dreams is, of course, to stimulate the autophagy. We will come to that. And finding, meaning stimulating autophagy, means that you have to find new molecule, new way to engage autophagy into the cell. The most common is rapamycin, is a star drug, very well used over the world, but we need much more. And I believe on the forest. My idea or my feeling is that hidden in the forest are molecules that from mushroom, from plants that can stimulate autophagy. And the idea is to give you a flavor here how you can search for this kind of molecule, how you can test natural extract into the lab to see if they have an effect of autophagy. And here is just a quick introduction and a schematic view on what is autophagy into the cells. Because some, you can say, molecules, macromolecules, aggregates that are floating in the cytoplasm of your cells get surrounded by a membrane called autophagosome, and this one is distributed to the lysosome where it's fused with the lysosome, and these particles here are degraded and they come again out to nourish the cells, to make new amino acid, new protein, new lipids, new copper, etc. I forget to say, if you have question, if you have comment, don't hesitate to interrupt me, because there is no bad question, there is just bad answer, so don't hesitate. And this field of molecular cell biology is not dogmatic. This is a permanent evolution. I see it since 37 years. Each year we have new theory, we see new stuff that make completely the one of the year before on the head. So don't hesitate to make a comment or a question or to ask me if something is not clear. The lysosome. The lysosome was discovered in the late 50s, beginning of the 60s, by a Belgian physicist, medical doctor called Christian de Duve. He gets a Nobel Prize for that. But for a long time, because a lack of tools in molecular biology emerged only in the late 70s, beginning of 80s. Also the new method for imaging, for biochemistry, and so he just observed actually the lysosome. He see it, and he see that this circle, that this sphere is growing in some condition, is reducing in some condition. So from that observation, the idea was transported for decades. It's just in the last few years that he changed that the lysosome is a kind of trash bag. It's a kind of sphere where molecules end, be degraded and re-export to the body, or a recycling bag. But nowadays, with the improvement of the molecular biology, of the biochemistry method, we know that this simple sphere is you can say a communication hub into the cells. It's integrate signals, macromolecules coming into the cells. It's looking what they mean, and it's also exporting some molecule. So it's an integration and an export. It's really a center of analysis from the cells. And here this guy, we will go later in more detail, is very important, mTOR. mTOR for mechanistic target of rapamycin. It's an example where a drug was able to show that a drug was able to show the function of a complex protein. And when this guy is inactive, it's floating around the cytoplasm, and autophagy can happen. When this guy is active, when mTOR is active, it's stuck to the lysosome, and autophagy is blocked. When a lot of nutrients are around the cell, into the cells, the autophagy process, the process of degradation of material into the cell, is not necessary. So mTOR block it. When you have starvation, when you have a problem, when you have not enough nutrient, mTOR is inactive, is released to the cytoplasm, and the autophagy process will happen. And I have this big chance that 2-3 years ago I was working on a project that was the first one that showed really this why mTOR gets stuck to the lysosome and why is not, why is more floating around into the lysosome. Of course, here you have just a summary. You have this role of degradation. This is quite clear because it's really in the center of the endocytosis and the exocytosis of the cell. But of course, you will see in the next slide, when you have a problem of lipid degradation also, you have also a central role of the lysosome. And last but not least, is regulating mTOR. The lysosome, when all is working well, you don't take care about it. But of course, once something is going wrong, then you have a huge problem. And historically, the first disease related to the lysosome were the so-called lysosomal storage disease, LSD, not the drugs. And I will go to an example in the next slide about this lysosomal storage disease. But if historically we were thinking this is the major problem when your lysosomes are not working well, in between, in the last 10-15 years, this central role for the lysosome emerged because, as you know, population get older and neurodegeneration get huge. Parkinson's disease, Alzheimer's disease, Huntington disease, all this stuff, dementia, when your brain goes wrong, is due to the accumulation into the cells of some aggregate. They are different for different diseases, but it's the accumulation of some aggregate. And they accumulate because the lysosome is not able to degrade it. And here you have the target that we identified by Novartis for some of them. Because of the power of the human genetic we have, we can check in patient which mutation is doing what. And of course, if these are the classical described, we find every week new one. even not in the gene, sometimes in the coding region of the gene. Of course, in cancer, the lysosomal dysfunction is also behind the progression of the tumor at the beginning, the tumoral step, but also on the progression of the tumor. Sorry. These are just two examples of the disease behind the dysfunction of the lysosome, because we know now that there are much more large problems if you get a problem on the lysosome. For example, your whole immune system cannot function very well. And all this field will probably increase a lot in the next decade. And here an example. You have this Niemann-Pick disease. It's quite rare, and it's a terrible disease because you have a progression all your life long. As you birth, you have some dysfunction, but also later it gets worse and worse. At the end, you end by dementia. What we find out, or what the people working on it find out, but a friend, Jürgen Reinhardt, was working on that, is that you have a transporter here, located on the membrane of the lysosome. It was called first NPC1, nowadays it's called SLC65. I will come to this family of the SLC because here also I predict that will be very, very important target for drug in the next future. If you have a problem, a mutation in this transporter, the cholesterol that is important to the cell and into the lysosome to be degraded for the constitution of new membrane when a cell is growing is not working well. So the sphaingolipid, the product of the degradation of the LDL, will stuck into the lysosome. And as you can imagine, when you don't get lipid, when you don't feed your cells with lipid in the next division, there is a huge problem because they cannot form the next membrane. Now we make a stop. This is one of my daily work. I just wanted to make you an examples of what I'm doing currently in the lab. And it's funny because this experiment I was doing just when Carla arrived to visit me for the first time in the lab. I make this picture. And what I make is I use a known protein of the membrane of the lysosome and I label it with some tag, hemagglutinin tag. This is a genetic construction. I use a gene for that protein. I add a linker that we have defined for another project that is that don't disturb anything into the cell, and I add what we call tag. Tag is just small epitopes that are derived from a virus, from the human influenza virus, so they are unique into the genome or into your human cells, because I forget to say we are using most of the time in the lab simply human cells in culture. And I express this genetic construction into different kind of cell through a lentiviral vector. And as you can see, with antibody, I can detect specifically this tag, and here you can see the lysosome. These are a high magnification of lysosome. This is a physical visualization of the lysosome. I tested on Western blot to show that the material that I get through an immunoprecipitation, meaning that I use beads, magnetic beads, that anchor this epitope, I can isolate, I can separate lysosome from the rest of our cells. And this is important because it gives you a possibility to physically isolate lysosome from the cell and to check what is on the membrane of the lysosome. Because as I say, we know that they identify each week new mutation. We can correlate it to core of patient and see, you have this mutation, you will get a dementia in 30 or in few years. Huh? But you have to verify if this mutation make that you have less of this molecule on the lysosome membrane, and then it's the transport of this molecule that is affected, or it's the function of this molecule that is affecting the transport function of this molecule. And for that, of course, you need to isolate physically the lysosome. This you probably know. Chloroquine, hydroxychloroquine, two major drugs, star drugs since the COVID. They block the progression of the malaria parasite and are used since decades. But what I find really interesting, they are derived from quinine, natural product. Is coming from a tree that is growing here. That is growing here in the forest and in the mountain of Peru. And what I find fantastic when you read about this quinine is that it's probably the first example of an ethno-pharmaceutical project or story. Because 2015, when the Spanish occupied Peru, when they arrived, they make some monastery, and in one of the monastery in Peru, the monks were ill. They have a lot of fever, and one Native American that was working in the garden of this monastery gives a powder of this tree into the monk, and the monk was healed. His fever goes down. They export this powder in Europe, and it was curing malaria from Italy, from Spain, where they were. So actually Native Americans save Europe. They make that Europe is Europe because malaria would have destroyed them at that time. It's important to notice this kind of stuff. But why I'm talking about quinine is, of course, self-interest, because we found in the lab there was huge debate during the COVID. Is quinine, is chloroquine, hydroxychloroquine working? It's blocking the virus or not? And of course there were big interest in Novartis. So they charged me to have a look on chloroquine, and we tested into the lab. And we see that it's a huge perturbation of the lysosome. I will come and explain you what are these markers, why they increase. But for the moment, you just have to believe me. You see a huge increase in this marker, meaning that your autophagy process is blocked. Lysosome is blocking chloroquine is blocking the function of the lysosome. Is a fantastic tools to study the lysosome. Now we move the circle will enlarge to the membrane. Because I have a patient for this kind of molecule. They are called transceptor. Transceptor. Words are very important. This is what I learned also from the Native American. Words build builds the world behind us or surrounding us. And transceptor is the combination between a receptor, a molecule or a protein that binds protein, and a transporter, something that transports molecule through the membrane. And transceptor, This word emerged from the plant for 20, 25 years ago. And now it's a field that is really expanding because we find several molecules that have this ability. It have the ability to bind a cilia and to respond to it, but it have also the ability to transport something. This is also very funny because this morning I get an email from home that this story gets accepted and it's in preprint in Bio Archive. And it's a huge story that takes us 2.5 years. We were about 40 scientists, researchers working on it, and we were paid from an European fund. Novartis give us also some money, and they give us the material, the laboratory, but a lot of money was coming from an European fund. And it's all about the solid carrier transporter, what it's called SLC. It's a new class of molecule, probably 300 at the moment that were identified, and they are transporter. You have a lot also, of course, of the membrane of the cell, the external membrane of the cell, but you have it also on the mitochondria, you have it everywhere. And I was in charge to see which one is on the surface, which one is on the membrane, in the membrane of the lysosome, and which one is involved in the transport in and out of the lysosome. A friend of mine, Fabian, is a proteomics guy. He is more a data scientist as a wet lab scientist. And with him, we perform some experiment in which we show the whole cell proteome in which protein is interacting with our specific lysosome transporter. And for that, I make kind of construct for several one in which I can induce through a drug the expression of this gene that is stick to a tag, so that we can see into the cell by microscopy where this stuff is expressed. And here, for example, we find out that this guy, this one, SSC 46, is really specifically expressed on the lysosome. Here highlighted in green. In red, they are marker of the lysosome to be sure that we are effectively on this organ. And this is the catharsis. This is the best examples of what is a transceptor and how it is working. As you see, I highlight the specific expression of this guy, SLC38, on the surface of the lysosome in green. It's schematically. And now, when you have low nutrient, this SLC is really compact on the membrane of the lysosome. It's like this, it's really a protein that is really compact. When it's a lot of nutrient, this is when no nutrient are available. I'd say nutrient, but actually it's all about arginine and amino acid. When you have low arginine, this stuff is compact. No? Autophagy can happen because you need arginine. When you have a lot of arginine, this guy is transporting arginine, and this compact structure get open, and you see here a kind of tile emerging. When this tile is emerging, a protein complex called regulator is coming to stick to this one, and this is necessary to bring mTOR on the surface of the lysosome, because the regulator is the receptor for mTOR on the surface of the lysosome. Very important, so I come again. It's really compact. When you have enough arginine, you don't need autophagy. When you have a lot of arginine, you have transport of arginine in this transporter. The tail is free. mTOR regulator bind this tail and it blocks autophagy. This is a mechanism that was showing, it makes a huge story, how the cells are able to modulate the function of mTOR. The next example I choose, because I choose just 2 examples, but we have of course a bit more, and with Fabian, but this is more a hobby, I'm not really paid for that. We are working on it to try to publish this story, to make the scientific community really aware about that stuff. But here for the second example, we have to step back in immunology. Very, very simple, my hand draw. You have a virus, it infects you, epithelial cells or endothelial cells. No? These cells infected by a virus, they don't feel good. They don't feel good, of course, and they start to die. And when they start to die, they send signal. They send cytokines. Actually, pro-inflammatory molecule called interferon type 1. And interferon type 1 is stimulating dendritic cells, in the sense that on the surface of the lysosome of dendritic cells you have this toll-like receptor that bind the DNA or the RNA of the virus, and they say to the cells, something is going wrong, you have to react. And this is exactly what's happening. Dendritic cells get activated, they start to eat the debris of the epithelial cells that die, and through that process toll-like receptor gets activated, because of course they also eat some piece of virus. They call for another one to help, because one dendritic cell, one macrophage will be not sufficient, so they have absolutely the need of friends that coming to help them to eat all the debris of the cells. And the process of an immune response, or when it's going very bad, an inflammation, is of course in process. And this guy is my friend, is Giulio Superti-Furga. And this guy, this is actually the guy that found the tyrosine kinase receptor when he was young. Now he's 62, and he found 2 years ago, and it was published last year, I think, the pathway that underlie this inflammation process. And of course I was also working with him on that. Why? Because again, a transceptor called SLC15A4 is involved. And the process we find is that RNA or DNA, or we can use also a drug, stimulate the toll-like receptor that in fact stimulate a cascade of kinase that phosphorylate a transcription factor only when this molecule that we find, TASL, bind to SLC15A4. And this transcription factor, when phosphorylate, dimerized. When dimerized, is transported to the nucleus. Pro-inflammatory molecules start to be transcribed, and of course, you have this problem of inflammation. And this is going further, this story, because in between, cryo-EM is also very fashion at the moment in Europe. I don't know here, but in Europe, a lot of invest is making this cryo-EM and structural biology facility. And this permits you to make the structure of all these molecules. And through that, we identify why and how tassel is binding on the surface of 15A4, starting the inflammatory process. Here we enlarge even more the circle. Now we finished with the membrane, with the specific transport story of the membrane, and we will go a bit to autophagy to give you a bit of flavor of what is autophagy and why it's so important into the process of health and disease. And as you can see here, people make mutation for this mTOR into Drosophila, and what's happened, the Drosophila is in life, but look the difference inside. This is a normal Drosophila. Here, the autophagy process is destroyed, is not working. So the growth of the Drosophila is quite reduced. Osumi, Professor Osumi, is the one that discovered and described the autophagy process in the beginning of the 90s, because as I say, the Christian de Duve that discovered the lysosome was describing a kind of you can say autophagy, because simply in the microscope he was able to identify the size, the lysosome growing or reducing. So he was expecting that something happened to the cell, a bit strange. But this guy, with the help of the genetic and the help of the yeast, modelized the autophagy at his molecular level. And what's happened is that you have as a first step in autophagy the formation of what it's called a phagophore. It's the combination of different proteins that form a complex that build a kind of membrane around the target that have to be degraded. This can be, for example, a mitochondria that is going wrong. Because when a mitochondria is going wrong, it will be destroyed, and this debris have to be eliminated. It's also the case for some protein or proteins that are not working well. So they will be moved in a circle called the autophagosome, and this one will at least fuse with the lysosome where this target will be degraded. This is the basic principle of autophagy. And this you find everywhere. You find it in very, very simple organisms. I just read, because I know that Professor Tavares is working with Chaga, that even the Chaga parasite, 2 cells, is using autophagy to survive and to multiply, because there is not enough nutrition for this parasite, so he will use this kind of process to survive and to grow. And as you can see, when you have no starvation, when you have enough food and everything, you can survive without autophagy. But as soon as you make a starvation and you have a mutation in one of these proteins that are necessary for autophagy, of course you cannot survive. It's really, really important. And here, rapamycin, as I tell you, rapamycin is really a star drug. Novartis would be not Novartis without rapamycin, because rapamycin makes Sandoz, and Novartis is coming from the fusion between Ciba and Sandoz. And rapamycin was found in the mid-60s, on this island, eastern island, or Rapa Nui in the native language. They say a Polynesian island, but in my mind is this island just facing Chile. And there was an expedition, and they find in the soil of this island microorganism, a fungi, and they purified a molecule that show antifungal activity. So they were thinking, oh wow, we have a new antibiotic or something like this, it's fantastic. But when they test it in people that have disease, there is a huge problem because because rapamycin is reducing the immune response. And you need the immune response when you have an infection. But at the same time, in the 70s, the first organ transplantation were possible. So reducing the immune system is fantastic because you block the rejection of organs. And from that on, rapamycin was used. And as you see, nowadays, rapamycin, you will find it in the literature, is used in many another disease as soon as you need a stimulation of the autophagy. Because this drug is the best one at the moment, really the best one. I would not say the only one, but it's not far, that stimulate autophagy. And it stimulates autophagy because it's blocking mTOR. This is mTOR, the mechanistic target of rapamycin. It was identified in the University of Basel as I was a master student there. And I was very jealous from the colleagues that were doing the master in this lab because they get famous. At the moment, they have started with this identification of the mTOR through quite complex genetic cloning into yeast, and they were showing that the mTOR, they use actually rapamycin to identify mTOR because some yeast are resistant to rapamycin, and through that they were able to find the gene that belong to this system. Here again, I wanted to show the way mTOR is working. When mTOR is inactive, it's floating around the cytoplasm. Autophagy can happen. When mTOR is binding on the regulator system, autophagy is blocked. So regulating mTOR is in fact regulating autophagy, when I want to be as simple as possible. And of course, rapamycin is regulating mTOR. It's blocking mTOR. Here, I wanted just to highlight how important autophagy is in a mammalian organism. And of course, for study that, mouse are beautiful. Because in mouse, you can destroy one or the another gene specifically. But when I was young and making what we call knockout gene, the problem in this kind of physiology is when you knock out a gene from the beginning, your animals cannot live. It's lethal. They cannot come to birth. But nowadays we have developed some methods, some tool that can make your knockout very specific in one or the another tissue, and that can be knocked out just at the moment you choose. And through this kind of complex homologous recombination in stem cell generation of transgenic mice, it was possible to really identify which gene of the autophagy is really necessary for this tissue, for that one, and so on and so on. And when we pass from the mouse model to the human, you can see here again in a very nice circle from the birth to the adult at which step, and at each step of the development, the process of autophagy is very necessary, because if you block it, if you manipulate it, you cannot go to the next step. And there is a huge amount of literature for each step. You will find to prove that your autophagy is really necessary from one stage, for example, in development to the next stage. And here I wanted to summarize all the problems you can have if your autophagy is not working well. And as mentioned, one of the major problems in the West is not the only one, but is a huge problem, is the fact that the people gets older, and this kind of disease, they increase a lot. And when your autophagy is blocked, you have much more this kind of problem. And we try, I will come back later on it, into the lab to use autophagy specifically to cure this kind of problem. But of course you have also cardiovascular disease, your pulmonary, your liver, you are not functioning very well, you have a lot of muscular dysfunction, and even of course the classical cancer, autoimmune disease, everything. Because this step, this autophagy physiology is on the cells and is in the center of the well-being of the cell. So as soon as it touches, you have a problem. Again, I wanted here to schematize a bit what is autophagy, the different steps, as you can see, the formation of this autophagosome, the fusion, and the molecule here, or the gene name that are listed here. When you have mutation, you block the step, one or the another step of the autophagy process. Meaning that when you have mutation in these genes listed here, you have a problem. But on the other hand, for us, they are possible targets to modulate autophagy. This is just an example of what's happened when you have a heart attack or when you have an ischemia. When you stimulate the autophagy, you have chance to survive. If not, of course, through the accident, you have oxygen deprivation, meaning that you have your mitochondria that are going not so well, and at the end, you induce more cell death, and your problem is much, much bigger. Huge invest by Novartis in this thing. It's using autophagy again, AIG. This is the ultimate dream of guys like Musk. They have everything, but they have a problem, they get older. So they want to survive longer, and that's why here there are a lot of invest in the use of autophagy to, not to cure, but to make the process of aging much slower. I'm working in a group, in a department that is called Human Genetic and Target. Simply because we are using all the power of the human genetic to identify mutations that are relevant for some disease in every type of disease. But of course, I'm using this power to identify new mutation in the case of autophagy, to try to see if I can find new possible intervention. An example that I get from the lab where we were using, which Huntington disease is a neurodegenerative disease. When you have a mutation, you have accumulation of aggregate into the brain. And we can reproduce this kind of disease in mouse. We overexpress this mutant in mouse, and by stimulating the autophagy, and normally in this, I forgot to say, sorry, we can reproduce this, we can express this mutation into mice, reproducing the pathology from the human into the mouse. When we make slides of the brain of these mice, we see degeneration of neurons. But when we stimulate autophagy, we see a rescue of this degeneration, meaning that you can use the potential of the autophagy to rescue neurons. And this is on what I'm working at the moment. And what we do is that we have some cells in which we express marker, fluorescent marker. And again, this marker, we have what we call nanobody. It's simply the V domain of an antibody. And this we couple to an autophagy transporter so that we can bring this molecule and degrade it into the lysosome. And this is working, it's fantastic. This is working, and we will try to improve this, because as you can imagine, when you have a pathology, you have your mitochondria, or you have an aggregate of neurodegenerative disease, like in Alzheimer, amyloid, or the synuclein in Parkinson, and we can bring tools to that and degrade it into the lysosome. This is my great hope, Professor Tavares, that we push this to the end, and that we can cure through this kind of tools all these problems. Maybe I have to accelerate because the time is running away. So we go to the last part, and the last part is how you can monitor it. For example, somebody brings you an extract from the forest into the lab. How you will look if it's modulate autophagy or not. And then there are several methods. You can use microscopy, luminescence, or you can simply do Western blot. So you don't need a huge equipment to do that. You have a basic. Will be a cell lab to culture cells, and a bit of biochemistry to do Western blot. With that, you are done. And here just an example of all the molecules that were identified as autophagy activator, that stimulate autophagy, like the example of rapamycin, and all the molecules that inhibit autophagy, like, for example, chloroquine or bafilomycin, the most common. And here, our ondro of the process, you have something to destroy, or you have an aggregate in the cells. Let's imagine you have synuclein that accumulate in a dopaminergic neuron of the brain in a human Parkinsonian patient. P62 is a transporter of autophagy, and this will bind this aggregate, and the complex aggregate plus transporter will stick to this molecule into the membrane of the autophagosome, and this permits the fusion. With bafilomycin, it's a drug, you can block this fusion. And when you stimulate your cell, for example, with an extract, you will see on the distribution of this molecule on a Western blot if it's modulating autophagy or not. So you take a cell, you stimulate it with a drug, and then you extract the protein of the cells, you put it on a Western, and you see if you have an effect on autophagy or not. Quite possible everywhere. Of course, you have another possibility, because you can label this kind of marker with fluorescent, and then you can see it directly into the cells if your extract is modulating autophagy or not. And this this is my own experiment, but it's exactly the same as I tell you. I wanted just to show that we are using actually this kind of method to see if something is stimulating autophagy or not. Betty was looking on natural extract. If a natural extract of a plant by us is stimulating autophagy or not. This can be increased and can be do much faster and better, of course, because you can use a fax if you have the money to buy a fax, but it's not necessary. And of course, if you have a big pharma company, then you increase to a high throughput with machines that do your work. And of course, you can screen for much bigger level, much more. But the simple cell culture stimulation, Western blot, you know if your extract is stimulating autophagy. With this, I wanted to finish and give you just a summary. As we see, the lysosomes are very central. They control the growth, the health of the cells. It's bridging really the metabolism with the physiology. We go to this transceptor concept, meaning that molecules are able to bind something but also to transport something. And they regulate the need of a cell. They are probably the most primitive example of, how we say in English, conscience. They are able to measure the environment and to react. Autophagy, I hope I give you a flavor how important this process, this physiology is in health and disease. And that's all. Actually, yeah. I wanted to just to finish with with some example, as it's possible everywhere in the world to monitor to see if something is modulating autophagy or not. This is my my colleagues. This is the lab. My boss, Beat Niefeler, the people I work with, Zach, postdoc in the lab. We make the tools together to destroy the possible target to autophagy. This is Fabian. He's a data scientist that generate all the work and the analysis for the solid carrier. This is Giulio, the boss of the Molecular Medicine Institute in Vienna. And these are two colleagues that I always ask for something, because first they are very nice, and they are much more mechanistic as me. They have under control all the screening possibility. And this is where I live, where I work. The institute that is located into the campus of Novartis. This is the center of the campus of Novartis. And of course, this is in the center of West London. So with this, I hope I convince you that autophagy is it's a very good topic to work on. And again, many thanks that I can present you all the stuff I'm currently doing. Thanks.
Thank you very much, Dr. Gilles, for this wonderful presentation. And is there any questions? Tem alguma pergunta? Doctor, I think we have any questions?
Deixa eu ver se consigo levar para ele.
Oh, here. Yes.
Oi.
Eu vou editar aqui, ó. Não, eu vou editar a câmera para ele.
First of all, sorry
I'm not a med student, so it's not going to be a topical question. I made some notes. Some may say we are in the modern era with the most pushback against science and scientists. Many scientists and science itself have been threatened and attacked. So, have you dealt with any pushbacks in your research? And for people who want to get into research, how to deal with backlash?
I think I don't really understand what you mean. Backlash? Backlash, you mean? Oh, you maybe say it in Portuguese so I can.
Pode falar em português.
Eu quis dizer que acho que a gente está na era moderna que tem mais ataques e repreensão de cientistas da ciência em si. Então, se você já teve que lidar com isso fazendo a sua pesquisa e como lidar com esse tipo de coisa para quem quer entrar em pesquisa, da credibilidade.
She said that in the modern area, it's common to attack to scientists, for example, repression and this kind of problem. Have you ever dealt with this kind of?
But what you mean with attacked? Because I was never attacked, and I'm working since 40 years. People is nice to me.
Not personally attacked, more in the sense of many people today don't actually believe in science. So, is that something hard to deal with people who don't really believe in what you're researching or the way people see scientists in general?
I think I see the point, yeah. You mean that the media or some people, they don't believe in science, they say we are manipulated by the money, we are manipulated by the government. Did you feel that I'm manipulated by any way? No, I'm not. Of course I'm paid by Novartis, I'm paid by the huge pharma industry since a long time. But I feel me free. I feel me free to talk to them, to say what I have on the heart, and I never get any problem. I believe that science is very necessary. I believe that we have to produce very hard data that prove the stuff. I think we have to prove, and Transceptor is a bit in this direction, that tree, animals, river, mountains, they all talk. We lost through the civilization this talk. We as scientists, we have the possibility to do experiments and to prove in the world, in the politician, that animals, plants, river, mountains, forests as to be important as the human itself. And I believe on the power. That's why I'm doing science, actually. I do science to prove in the politician that what the Indian in the forest say is true. Because an Indian, he say that when you ask him why this plant is good, he will tell you, yeah, because the plants tell me. And this is true. The plants tell him. We just don't have at the moment all the methods to prove that the plant can speak to the Indian, but it's the case. 2 weeks ago I read that in Brazil some people make, because you have fantastic research here, some people make a kind of captor over the tree into the forest, and they see molecules coming out of the tree that signal in the sky that it has to rain because it's dry. So the plants are talking to the sky. This is what the native people say. We can prove it, and that's why I'm doing that.
Do you have any advice for people that want to enter in research?
Be patient. believe on what you do, work a lot. Like Professor Tavares, work day and night, 2 o'clock in the morning, then you will be a scientist.
Good question. How is that lysosomal dysfunction related to metabolic diseases? And how can this understanding pave the way for new treatments? I ask this because I search about diseases, diabetes, atherosclerosis.
So this is a very good question, but very, very difficult. It's very, very difficult because when you talk about metabolic disease, it's a family of disease, a lot of stuff. But for example, in some metabolic disease, you have accumulation of some protein, of some macromolecules into the cells. And autophagy will help the cells to degrade this accumulation, for example.
Hi, Dr. Gilles.
Hi.
I'm Lillian.
Nice
And
to-
I have a question, but just for curious about, you were talking about the lysosome extraction in the sample.
Yeah.
And how difficult is to isolate lysosomes from samples? And is it a stable process?
It is, yeah. I'm working on to be. I must say honestly, I'm working since a year on it to make it really easy. And it's not the case, because in the past there was a possibility to use centrifugation step to separate and differentiate organelle into the cells. And now, through the power of the genetic, we can label the lysosome and isolate it. It's quite dependent on the cell type you choose, but it's possible to do because I do it since 1.5 years and it's really working. We are able to isolate lysosome. 2.5 days. 2.5 days maximum. You probably probably at the end of the year, I hope, because it's in my objective, I can reduce it to 2 days. But you have an overnight step. You have an overnight step that I feel you cannot really reduce. Yeah. Thank you. It was a very nice question.
Hey, Dr. Gilles, I'd like to thank you for the talk.
Thank
It's
you.
a very interesting thing. And I'd like to know about the markers. You show some markers.
Yeah.
And those markers, I can investigate them in cell culture.
Yeah.
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 And I have to investigate all of them to have to know if it's occurring autophagy or not.
Yeah.
All of them.
Yeah.
I can't just choose some of them.
No, if you want to follow the process of autophagy, it's better to use several markers. Yeah, because it's also this makes the story very difficult. For one cell type, you can say you have an increase in marker X and a reduction in another one. But for some another cell, you have an increase in both of them. So you have to check for each cell. First, you have to have a model. You have to choose a cell, a HEC or a GHP1. And then in this cell, you check autophagy. And you use all the markers. And then you will see if one is increasing, if one is decreasing. Yeah.
Okay. And now then I can check by Western blot.
Yeah, you can check by Western blot. Yeah.
All of them.
Yeah, all of them.
Thank
All
you.
of them. All of them.
Dr. Gilles, thank
Yeah.
you for your lecture. I learned a lot.
Thank you.
And I thank you because you showed us it's easier than you think than I think to study autophagy.
Yeah.
It's related with a lot of diseases.
Yeah.
I'm trying to be a neuroscientist
Yeah.
and a cellular biologist.
Yeah.
And we are here in the forest,
Yeah.
as you said. Do you have any tip what kind of molecules we can start with? You showed it us, but could you talk a little bit more?
If you ask me, I would directly start with acai. Directly. Because acai, what I learned, it was Friday evening, we have a party with that organized Professor Tavares. And outside, we were smoking a cigarette with Abraham. And Abraham told me that acai came from the Native American, from the Indians. It's a legend because a baby was suffering, not enough to eat, and a grandmother asked the forest to help. And the forest gave acai. And acai saved the life of the baby. It's what he said, no, we know. And acai, of course, is also full of calories, but maybe it's stimulating autophagy. And this I would look. If you ask me an example, but there are many, probably many. I was imagining, and that's why I'm here, because Professor Tavares promised me to take me to the native, into the forest. And this will be the experience of my life, because since I'm French, I'm Alsatian, but I grew in a countryside, in a valley, but I don't know why. Since I'm like this, since I can speak, I was fascinated by the Native American. And so it's really the dream of my life, and I never met anyone. And yeah, this is really the dream of my life, to meet them, to see how they behave, to see how they walk in the forest, how they observe things, and I'm sure that the connection with these people will help us to find which plants are necessary for which disease, and especially in this case of autophagy, because autophagy is behind every disease actually. So when you find something that modulates autophagy, probably you can act on each disease. And I think Native American, if we don't listen to Native American words, we cannot continue. We are destroying everything. With our rationality, with our civilization, we destroy everything. Native American, they know that you have to respect the blood of grass as every human being. And if we don't listen to them, we cannot continue like this. This I'm convinced. That's why it's very important to listen to them and to work with them, to get the spirit of them.
Hi, I would like to know what are the next steps of your research, and if are there any worries about this mechanism, the autophagy, affecting healthy cells, because you aim to degrade.
Yeah, this is a very good question.
Yeah.
This is a very good question.
Like side effects of drugs.
Yeah. Okay. Sorry. This is a very good question, because life as a phenomenon is really subjected to homeostasis, meaning that you have a balance in everything. If you push too much the autophagy, your cells are dying. So you have to find the real, how you say, this balance of inducing but not too much. But this is for every drug, it's for everything. Even sport is very healthy, but if you do too much sport, you will have a huge problem.
Hi, Dr. Gilles. Just for curious, what are your expectations when researching Amazonian plants? And have you read about any specific species of plants we have here besides acai?
No. No, I try. Since I met Professor Tavares in Joao PhD, I'm reading about Amazonia, but no, I'm not a botanist. Definitely not. I'm a pure lab rat. I'm since years in my laboratory making experiments. I don't have this knowledge about which plant is doing what.
All right, thank you.
But you are the guy that knows much more about that, and science is a question of collaboration. Nobody can know everything. I think you have to be you have a kind of expertise, but you have to be open to your colleagues, to your friends, listen to them, and try to incorporate every knowledge to it. Yeah.
Doctor, we have a question from Abraham Victor Tavares. He is asking, can the epigenetic alteration interfere in the genes that are involved in lysosomic disease?
If the epigenetic...
Can the epigenetic alterations interfere in the genes that are involved in the lysosomic disease?
Very good question. I don't know, but I don't know. I have to check. But epigenetics regulate the state of your genome. I know colleagues that are working on epigenetics. They tell me the histone, the molecule that compacts the genome into the cells, are affected by epigenetics. Of course, all these genes that are used in the autophagic process are under this control. So, I mean, if you have a factor, an epigenetic factor that influences your genome, of course, it will influence your autophagy. But I don't know. It's just a reflection.
Okay, thank you very much. I think that's it. Is there any question left? Thank you very much.
Thank you for your attention. It was a pleasure.